Good afternoon, Year 12. So, we carry on with this topic of DNA, genes and chromosomes with a lesson on transcription. Now, this relies heavily on having learned the RNA subject, which we learned in the last lockdown. So I want you to have a look at those three questions, which are on the board, which is basically a starter for this lesson to make sure everyone's up to the right point. So you have a couple of minutes to do this. The most important bit is the drawing of the monomer and knowing what the different bits are. So, as a reminder, you should be pausing your video when I'm asking you to do a task, because we're trying that way now. Hopefully you did. Hopefully we should know that the monomer that makes up RNA is a RNA nucleotide. A nucleotide is just fine. Draw this monomer. Now it's exactly the same as a DNA nucleotide in terms of look. You want a circle for a phosphate. You want that attached to a pentagon, which you're going to call ribose, and that's the main difference. In fact, the only difference for now is that it's ribose and not deoxyribose. And then you want that it's attached to a base. Now that base can be adenine, guanine, cytosine, or, and this is the second option, uracil. Uracil instead of thymine. How are they connected into a polymer? Well, they're connected to each other via a phosphodiester bond, and that involves a condensation reaction. So if you got that bit, then you're up to speed. If you didn't get that bit, I would go back and look at the RNA lesson, which is still on YouTube, have a look through that, make sure you've got RNA, because it is important for this topic. So once we've got that, let's have a quick look forward. Today we'll be talking about how RNA is made. Now we're going to start off by talking about another kind of RNA which we need to know about, called tRNA. But the main point of the lesson is to learn about something called transcription. And this is the first stage in protein synthesis, but this is how we're going to make RNA. So by the end of the lesson you should be able to do things on your screen now. I'm going to start off like this. So for now there are two main types of RNA which we need to know about. We learned about one of them in the last lesson on RNA, which is mRNA. So when you're thinking about your long line-like strand of RNA with all the monomers attached together in a phosphodiester bond into one long line, that is mRNA. And mRNA is, to be fair, I'm answering your question for you at the bottom there. mRNA has one function, but there's another one called tRNA, which is a completely different function. So you should be able to describe the structure of an mRNA molecule. Hopefully, everyone understands that mRNA is made of nucleotides attached by phosphodiester bonds into a single chain. So we're not talking about a double helix, just a single helix with bases sticking out of it, either adenine, guanine, cytosine, or uracil. Now, mRNA is used to transport the genetic information from the nucleus to the ribosome, where we're going to make the proteins. Now, those ribosomes could be in cytoplasm, they could be free-floating, or they could be in the rough endoplasmic reticulum, on the rough endoplasmic reticulum. However, there is another type of RNA which is going to be essential for the next couple of lessons, called tRNA. Now, you kind of came across this in year 11. We called it a carrier molecule back then, but your teacher may have used the word tRNA. And tRNA looks like what you can see there. Now, you don't need to be able to draw that much detail. We don't care about the acceptor stem and the T arm and the D arm. All we need to be able to say is that it has a cloverleaf structure. Now, if you follow it around with your finger, you can see that it is still one chain. So you've got the blue bit, then the orange bit, then the blue bit, then the purple bit, then the blue bit, the green bit, and then the blue bit again. But it's all just one long chain. But every now and then you find that the chain folds back on itself and the bases form hydrogen bonds, forming this cloverleaf structure. Cloverleaf because it's got three leaves, if you like, attached to it, like a clover. So a very common question is to describe the structure of tRNA, and you just need to be able to say it's a single chain of nucleotides, RNA nucleotides that form a cloverleaf structure. I'm going to talk about what it does in an enormous amount of detail next lesson. For now, all I need you to know is what's on the board. So if you haven't got those notes down yet, please pause the screen and make sure you get those notes down. Well, incidentally, if you haven't got the title yet, it is transcription. Transcription. And that is on the previous slide, it'll be on the next slide as well if you want to leave it blank. So, once you've got that, we need to be able to answer these questions. Now, this is just about the structure of DNA, mRNA, and tRNA. So have a look at that. I'd like this drawn into your books, and that will take a bit of time. I'd like you to tick underneath DNA, RNA, and tRNA what facts you think are true about it. So if you think that DNA contains the uracil, don't, but you would tick DNA contains uracil. So I'll give you 3 minutes to write that down. Please pause the video and fill it in for yourself. So, hopefully you've done that already. We've got DNA, mRNA, and tRNA. So, made of nucleotides. Made of nucleotides is DNA? Yes, DNA is made of nucleotides. And so is RNA. So mRNA and tRNA are made of nucleotides too. Contains ribose. Only RNA contains ribose. You may think that deoxyribose somehow contains ribose and a bit of deoxy. I've heard people done that before. No, it's a different sugar. So DNA does not contain ribose. Double-stranded? Only DNA is double-stranded. And another misconception is that tRNA is double-stranded because when you look at the structure, it kind of looks here at the top, not sure if you can see my mouse, that it looks like it might be double-stranded, but it's still just one long strand that's folded back on itself. For a double-stranded thing, you'd need two separate polymers, two separate chains attached to each other. This is one long chain. So only DNA is double-stranded in this case. Found mostly in the nucleus. Now we haven't talked much about the function of tRNA, so you may have ticked that one. It's not found in the nucleus at all. Neither is mRNA mostly found in the nucleus. DNA is the only one that is mostly found in the nucleus. Contains uracil. Uracil is only found in RNA, so that is mRNA and tRNA. And then has an anticodon. Now this is just to see if people are paying attention. We're going to come back to this anticodon in a lot of detail later on. The anticodon is used when tRNA is used in protein synthesis. And it's only found on tRNA, not on anything else. So tRNA has an anticodon. And quite a nice answer for describe the difference between mRNA and tRNA is that the tRNA has this anticodon. And the anticodon is just these three bases at the bottom of that little pink bit that stick outwards. All the other bases stick inwards, those three stick outwards. And they are going to be essential when we talk about protein synthesis next lesson. Now, that's it so far for mRNA and tRNA. That's all we're going to do for now. We're going to use that in a lot of detail next lesson, so keep that. The next bit we're going to do is we're going to move on to protein synthesis proper. Now, you need to be able to describe how we can go from a long strand of DNA to a protein. We did a bit of this in year 11, but we're going to do a lot more detail in A-level. It's pretty essential for life. If it goes wrong, you get all kinds of horrible cancers and nasty diseases. And so we're going to learn about it in a lot of detail. Now, what I'd like you to do is I'd like you to have a think back. I'd like you to, in a second, when I stop talking, pause the video and just think, what stages of protein synthesis do I already know? And I think you may surprise yourself. You may know more than you think. If you think, actually, I can't remember anything really, then that's fine. We're going to go through it. But I just want you to think, what do I already know? To give yourself a framework. So have just a minute. What do you already know? Pause the video. Right, so what we should already know is we should know that DNA is used to create this molecule, which is called mRNA. That mRNA then goes to the ribosome, where a carrier molecule brings amino acids to the mRNA, and it's used to then create proteins out of these amino acids. And that's about as much detail as we did in GCSE. Let's go into a bit more detail. Now, the general A-level overview for protein synthesis is, firstly, a free mRNA strand is created using the DNA as a template. And that's called transcription. And that's the stage we're going to focus on today. That then leaves the nucleus through a nuclear pore and is spliced, and we're going to talk all about splicing today as well, into something called full mRNA. That then goes to the ribosome, where it's read to produce polypeptides, and that is called translation. And we're going to learn all about that next lesson, as I've said. And then it's then folded into a protein. So that's just a general overview. That's the general idea. And if you don't know that, to be fair, I wouldn't expect you to know, particularly steps 2 and 3 there. Can you get down that overview now? Because it is important. So again, I'll give you 30 seconds, just pause the video. Right, that's the overview, but we have to know more detail than that. If you feel like you're lost already, we are going to go through each stage in detail, so please don't worry. Let's start with transcription. Now, transcription is how we go from DNA to pre-mRNA. Basically, we've got our DNA, which is our double helix, our massive, enormous, long chromosome molecule, and we've turned it into a very short piece of mRNA, which codes for one protein. It's from one gene. So we unwind our one gene. Let's say I'm creating a protein in my eye coloring protein in my iris. I unwind my eye color gene, and I'm going to create some mRNA, which is basically going to be used to take the information about my eye color to the ribosome, which is going to make the protein. So you can think of the mRNA as a messenger. That's why it's called messenger RNA. It's taking the information from the DNA and it's saying to the ribosome, here's the protein you have to make. So we have to make that mRNA. And again, there's some stages here. So let me talk you through them and then you're going to write them down. And you're going to notice that this is a lot like DNA replication. In fact, it's pretty much the same process with a couple of extra bits. Firstly, you unwind and separate the DNA helix with an enzyme, which we're going to call helicase. Now, you remember that enzyme from DNA replication. It's actually a very slightly different helicase. There are lots, but we don't need to know anything about that for A-level. But we unwind and we separate the helix. We break the hydrogen bonds between the bases, just like in DNA replication. And in actual fact, on that first step, I would like you to write down that hydrogen bonds are broken there, because that's quite important, and I haven't included it on the side. Then, you're going to get the nucleotide bases on one of the strands, pairing with the complementary RNA nucleotide bases that are floating free in the nucleus. So in DNA replication, what we did was we unwound the helix, we broke the hydrogen bonds, and then free nucleotides